Greetings. My name is Wesley Straub, Marketing Director here at Repligen, and welcome to the second episode of our 2022 webinar series. In the first episode, we introduced a new platform for development of custom affinity resins with breakthrough success rates, timelines, and performance. Today, we're going to stay around the chromatography unit operation and introduce an inline analytical technology that can be applied to the chromatography step, but also be applied to other downstream unit operations such as TFF and formulations, right? Today's speaker, Ramzi Shambaki, Director of Bioprocess Applications, has been an integral member of both the internal technology development team as well as directly working with customers in the field on developing applications. So we're really lucky to have him because it allows him to tell the story with a really unique set of breadth and depth on the subject. So because of all that, you don't have to hold back in any way on any other questions. As always, we have a few housekeeping items that we need to take care of. The first is that this webinar is being recorded so that it can be viewed by those who could not join us today. Second, we have a little bit of interactivity in the platform. On the side panel to your side, you'll see that we have a poll question. We ask for you to take 30 seconds and provide us with one answer to that poll question. It really helps us connect the content of these webinars with your interests. There's also, as you'll notice, a Q&A panel for live Q&A right beside the poll question. You can ask a question at any time throughout the webinar. We will answer them in a live session at the end. If you like a question, we ask that you give it an upvote. We don't have typically have more questions than we have time to answer. And if you upvote it, we'll prioritize those. If we don't get to your question today, we will provide a written response within 24 hours. Lastly, if you'd like to chat with Ramzi, and we all like to talk shop with Ramzi, you have 3 ways to get a hold of him. You can send him an email. We'll post his address at the end. You can respond to an email that will be in your inbox immediately following this webinar, or you can scan the 2D barcode that is present throughout the presentation. All right, so with that, I'm going to turn it over to Ramzi and his talk entitled Realization of PAT Inline Using Variable Length Spectroscopy Applications in Downstream Monoclonal Antibodies, Viral Vector, and Oligonucleotide Purification.
Thanks, Wes, for the intro. Today, we're going to talk about the FlowVPX system and how it can help your biologics process development and GMP manufacturing. First, we'll go through some features in the system, where it's used, and why we need process analytics, how it helps the process overall. Then we're going to go into some detail on how the method works, both for the FlowVPX, FlowVPE, and SoloVPE. And then we'll have a set of data from various process steps on where the FlowVPX has been used and the benefits of each. A little intro to Repligen. We're a bioprocess company, and we have a various suite of products. Today, we're talking about the analytical solutions, specifically the FlowVPE and FlowVPX. But we have products that range from the upstream to the downstream. Things like pre-packed columns, single-use systems, ATF for cell retention, and various small-scale downstream systems. How the FlowVPE and FlowVPX interacts with those things will also be discussed. So this is an image of a typical bioprocess, starting from the early cell culture all the way down to the downstream. There are various locations where you can bring the FlowVPX into your process stream that provide some benefit. Typically, we are mostly in the downstream. So starting after harvest, our first application will be discussing capture chromatography and the benefits of that. And then we'll go into some discussion about TFF and UFDF on the downstream and general filtration. And then how the FlowVPX can be used in the drug product process. So why do we need process analytics? How does it help your process? In the current state in manufacturing, we have a very constrained process. So there are raw material inputs, say your cell culture. It goes through this constrained downstream purification process. And then you need to use various instruments to monitor the variable product quality attributes. There can be a wide range of those product quality attributes that leads to a certain consistency of your product. In the future, what we want to do is bring some of these analytical tools into the process. When you do that, you can actually adapt the process based on the information that you receive from these analytical tools, allowing for a more predictable product quality attribute or a tighter control of your process in the future. So how do you do that? Implementing a PAT is a complicated, long process, but it typically starts in the process development lab. You would get the instrument, you would bring it into your lab, and you'd do evaluations on various process steps and use that to understand your process better. Once you've done that, you can then zero in on a specific process that has a great benefit, bring that into, say, clinical manufacturing or some kind of manufacturing upscale, pilot scale, something like that, and use it to remove some sampling steps or add monitoring to the process that provides more information in real-time actionable information. Finally, in the final stage of this, you would take that product or analytical tool and you would connect it to the process, add it to your automation, which then adds it to the process control, realizing that adaptive process control and giving you that predictable product quality attribute. So how does variable path-length spectroscopy work? We'll go into some detail on the scientific method. Here you can see an image of two different types of absorbance spectroscopy. On the left-hand side, we have the typical uv-vis spectroscopy where the light is shined through a cuvette and it's collected on the other side in a detector. There, the path length is fixed. You're stuck with a 1 centimeter cuvette, and if your concentration goes out of the range of the instrument, you're forced to dilute the sample, and it requires some analyst to do that work. In variable path length spectroscopy, we've unlocked path length. The light now shines through a fiber optic that we call the fibrette. The distance or the path length is defined from the bottom of the vessel, or in the case of the flow VPX, the bottom of the flow path, up to the tip of the fibrette. The fibrette is attached to a precision linear stage that allows motion of the fibrette in the sample in real time and collects an absorbance versus path length plot. That relates directly to your concentration by dividing by your extinction coefficient. So rather than having to do all kinds of analyst dilutions and making sure you have matched cuvettes and baselining and doing these things, you can take a direct measurement with the solo VPE or the flow VPE and get your concentration immediately with essentially a simple push of the button. This makes it suitable for manufacturing, for IPC testing, and those environments. So the flow VPX works very much the same way as the flow VPE and the solo VPE. It's taking a number of data points in real time as the sample is running through a flow path that's attached to the base of the instrument. You can see a picture of the instrument here on the left-hand side. And what I like to say is that in a minute, you can generate 12 QC grade concentration measurements. So that means every 5 seconds, you're actually getting a full path length versus absorbance plot for each concentration data point. With those, you'll get an R-squared value, and that R-squared value tells you the linearity of your measurement. So if something like a bubble passes through, you'll see a drop in R-squared, which then the scientist or the operator can look at that and say, okay, this is a bad measurement because the R-squared dropped. So for the good measurements, your R-squared will be 0.999 or higher. Let's go through some of the features of the system. We'll have a kind of high-level look here to give you an idea of how this system can be implemented in your process. So like I mentioned before, the goals here are to increase your process understanding initially, to reduce the cycling time and also maybe remove sampling steps, and then ultimately strengthening your process control, which will minimize the risk of your process. If you have a tight process where you need to hit a high concentration target, you'll be able to do so accurately with this system without having to take a lot of sampling. The system is made to be GMP grade. All the components are 316L stainless. Those come with certs that we can provide so that you can bring this to your QA group and file the information. The system has a vast concentration range. It can go over 300 mg per mil for a typical antibody. Low back pressure, as you can see from the image in the center, the fibrette just kind of sticks a little bit into the sample and doesn't present a back pressure issue. And it can be mounted easily with some screws in the back. And then all the connectivity options are available to connect to your external systems. So a typical simple analog 4 to 20 milliamp is available, some digital channels, and OPC UA, which would connect to Delta V or Siemens SciPad or those type of systems. The FlowVPX is fully scalable with the head unit that you would get. You also get a choice of 2 types of flow cells. They're interchangeable. We have everything from a 3 millimeter to a 1-inch pipe size. The 3 millimeter is typically used in the lab scale on the benchtop experiments. Then if you get the 3 millimeter and the 1/2 inch, you can take that same unit, bring it to your pilot scale, do processes over there for comparability, and then you can even scale up again with the same unit to the 1-inch pipe size for production scale. Coming later this year, we'll have single-use flow cells. Those will be available either irradiated or not, and then they also fit onto the FlowVPX unit, the existing FlowVPX unit. So you can move to a single-use environment if you so choose. For those who are familiar with the FlowVPE system, our predecessor of the FlowVPX, the FlowVPX was a new system that we came out with last year, and we made some significant improvements both in the size and the concentration range and the connectivity. So the FlowVPE system, though very capable, had a limited concentration range, which we no longer see with the VPX with a very high concentration range. It also didn't have all the options of connectivity like the FlowVPX does. FlowVPX, like I mentioned, has digital, analog, and OPC UA. The other factors that help in the VPX system for GMP adoption are the IP rating, it's IP65 rated, and the C1D2 rating, which allows you to put it into environments like peptide manufacturing, where you might use methanol as a buffer and need to have that C1D2 or ATEX rating. So here's where the rubber hits the road. We're going to go through some technical information and data. These sets are all from our customers. Our customers allowed us to present this information, some of them with mention here. And we'll start with chromatography. So from the early days of using the FlowVPE with some folks in continuous manufacturing, we discovered the possibility of using this system to measure titer in a protein A capture step. In order to do so, you have to read the concentration of the harvested cell culture fluid coming through the unit directly, and then either with a column bypass or a separate system, measure the concentration of the column effluent coming off the protein A column. If you do that and measure the difference between those two signals, it's equivalent to the titer of the process. This is because protein A is a very good, a very strong ligand. It captures the antibody very well. So you can get this analytical method to measure the titer. Once you have the titer, you also have the time and the flow rate, which will give you the mass, integrated mass of the process. And this could tell you what the loading mass is on your protein A column. And also, you can directly measure the elution mass by multiplying the concentration by the flow rate during the elution stage of the process. So this is the basis for some further work that we did in development, which I'll show on the next slide. So that method I mentioned before is the basis for being able to measure dynamic binding capacity. So KBI used the FlowVPE in this method to measure the dynamic binding capacity of various resins for various molecules. The advantage of this is that you can use your real harvested cell culture fluid. You can do it with experiments that you were planning for some other method, and you don't have to do a separate experiment for this. And with this technique, you can quickly see the impact of various resins with different molecules and how the dynamic binding capacity could change, which will definitely help your development. I like to point out on the line in gold, bispecific monoclonal antibody 2. It turns out with one of the resins, they got a 51 mg per mil or gram per liter dynamic binding capacity, but another resin only gave them 35. They could quickly identify that and say, look, okay, we're not going to use this other resin because we have a higher binding capacity with this one. Similarly, you can use the same technique to look at other features or other factors in the process, like different flow rates, different resin lots, how long you've reused it, lifetime of the resin. And each time you can see the dynamic binding capacity here. So you can use it for screening the resins and understanding them very well without having to send lots of samples over to your colleagues in the analytical labs. ABI released a paper earlier this year to outline their work. It's available on the link here. And the quote at the end that I'll just read it out is, the data shows that the resolution of this method is better than the traditional HPLC method, which that to me is very important. Using FlowVPE for DBC assessment during the capture purification step saves at least 2 weeks of analytical testing time and cost and eliminates downstream process development idle time, meaning you don't have to wait for those samples. You can get the job done on your own without sending samples off to the analytical labs, and you're saving your colleagues in analytical a lot of time for these measurements. We've done similar techniques in other modalities, so not just in the mAb space, but in the oligonucleotide space, you can do the same thing. And because the FlowVPX has such a high concentration range, we were able to read an elution peak off of HPLC in a ASO process over 75 mg per mil. If you try to equate that to the value from an antibody, it'd be similar to something that's nearly 1,000 mg per mil. We did this using the direct measurement at 260 nanometers, so the peak of that particular molecule, rather than using a surrogate wavelength like 295, which might present some error. In this study, we also took the integrated mass of the pool as it was being collected and compared it to offline measurements. And you can see at the bottom here, the difference is only 2.9%. So rather than taking a sample and measuring it offline, you can do this in real time as the product is being purified. So I'd like to move on now to another step in the process, the UFDF or TFF. The FlowVPX can be used in this process similar to what this diagram says. We like to put it directly after the feed vessel, because in that case, the product is homogenized through mixing in the feed vessel, and you can get a very accurate measurement of the product pool on the FlowVPX. The only place we don't like to put it is directly after the pump. It can cause some pulsation depending on the concentration range in the product. That can give you a noisy measurement. So if you put it on the suction side of the pump, you'll get a very accurate measurement of the pool. So breaking down the process here a little bit to show you guys the limits and what the advantages are. We took 3 systems and compared them all 3 of our systems, the FlowVPX directly in line with the FlowVPE at the same exact time. And then we took samples at various steps of the process and compared it to the offline SoloVPE. In UF1 here, you can see it's relatively uneventful, up to 50 mg per mil. All the systems are agreeing, and the limits in the red lines are shown to show that the target concentrations that they're using for the next follow-on step. In the dye filtration, it's somewhat similar. This was done manually. So in the dye filtration, you can see it kind of gets close to the limit in one location, but all 3 systems are very well in agreement throughout the process, holding steady at about 50 mg per mil. So Finally, in the UF2, the final and most critical stage of the UF, this is where some action happened. With this particular system, we were doing the test in a pilot plant, and it was on I would say a little bit larger scale system. They were running the final endpoint based on the volume of the system. Because of that, even though they were targeting 225 mg per mil, the system stopped, and you can see the plateau at around 170 mg per mil. Because we had the systems in line and the FlowVPX was giving an accurate concentration, the operators were able to identify that and see that the system had stopped early. And this was due to a miscalculation of the hold-up volume of the system. So then they manually ran the system up to 225 mg per mil. Once they got the measurement on the VPE and VPX, they took a sample off to the solo VPE and completed the process. So this was a perfect example of how you can do endpoint control with UFDF and the FlowVPX. And just a final table here to show the accuracy of each system and the comparability of each system. So knowing this, in our internal labs, we decided to make an automated UFDF system ourselves. We took the FlowVPX and connected it to the new Repligen FS15 TFF system. There we connected the KFCom software to our Viper software, and we automated the process based on concentration. Here's a snippet of data that you can see. The sort of manual, it's not manual, it's automated via the weight in the blue, and then automated via concentration in the yellow. On the first side, on the left side, we are targeting 20 mg per mil. And you can see we hit the target exactly with our concentrated automated system. Whereas by weight, it was a little bit off by a couple mg per mil. And then it gets even worse at the higher concentration. So by weight, we were targeting 100 mg per mil. You can see in the yellow, the signal floating around 100 mg per mil, but in the blue, it was bouncing around 110 mg per mil or so. So having the concentration control allows you to get a more consistent process and more accurate to the targets that you're looking for. So once you've completed the process and you're looking to recover your protein, it's often practiced to flush some buffer through the system to capture every last drop of that valuable antibody. This was done at a customer where they put the FlowVPX in line during this recovery process, and you could see how it slightly dilutes the process. So they want to be sure that they don't go out of their target zone, which they did here, hitting 200 mg per mil pretty closely. So it's not just proteins that you can use this system on. In fact, you can use it on AAV, AV, or other modalities as long as there's some UV absorbance that's available. In the case of an AAV, you have the capsid absorbance, and then you have the payload absorbance, the DNA. So it's a bit of a calculation, but if you get the slope value from 260 and 280, you have the extinction coefficients of both the 260 and 280 for the capsid and the DNA. You can calculate the VG per mil happening in real time. Here you can see a process where we started at around E11 and went up to about E12, and then did a difiltration near the end at E12. It is a bit of a noisy signal. This could be due to the material itself, but at some point, when the scientists looked back at the mixer and the feed vessel, the vessel was getting cloudy, and it corresponded to an increase of the signal from the FlowVPX. At that point, we decided to slow down the mixer because we thought maybe the agitation was causing some issue, and you could see the signal drop right there around 70 to 80 minutes. At each stage, we took a sample and sent it off to the ddPCR. Unfortunately, the first sample was a little bit off because we had already sampled before we started the process and it might have concentrated slightly. But you can see that even with that, all the values were very close to the offline ddPCR. So I think this is a pretty good proof of concept to show that it's possible to use this system for the concentration step in your AAV process. Finally, we'll talk about the drug product. So after you've completed your bulk drug substance and you've used the FlowVPX to hit your target exactly, someone else is going to take that DS and they're going to dilute it down and put it into vials or syringes to go off to the customer. In this example, the FlowVPX is put in recirculation loop with the formulation tank, and the target concentration is hit by monitoring the concentration as it's being diluted. In this example, we go from 90 mg per mil to about 5 mg per mil. You'll stop and recirculate when you hit the target. And if you'd like, take an offline sample, or you can use this to connect to your automation and get the target exactly during the automated process. Another area in the drug product that this has been used is to evaluate the sterile filters. Various sizes of sterile filter can hold up some of the protein, and it's not necessarily linear. So the smallest size you can see in the gray line only holds up a small amount, but the larger size in the blue line requires a lot more flushing to hit the target concentration prior to filling. From this, you can directly save materials in your process by optimizing that concentration curve that you see there. The FlowVPX comes with Viper Analytics software, and I'll just touch on it rather quickly. Viper Analytics is our platform for all our SoloVPE, FlowVPE, and FlowVPX instruments. Here you can see a screenshot. It's fully LDAP controlled, so you can log in and then you'll go into a specific app, either for the SoloVPE or the FlowVPX. It's intuitive, it's easy to use, it's app-based, it has all the 21 CFR Part 11 controls available. You can manage groups, you can manage granular control of each individual item on the software. It has a validation check package as well that you can do all the suitability tests that you need for the system. And it's ready to be integrated. It has OPC UA built in, so we can send you a node list of all the different nodes that can be available on OPC, and then you can use it to connect to all the external softwares like Delta-V. I mentioned it above, but I'll quickly mention it here. The FlowVPX is ATEX and C1D2 rated, so it needs to be for things like the oligonucleotide processing that I showed earlier. So you can put this in that kind of environment and feel comfortable, and we can provide the certs accordingly on your request. All the flow cells are cleanable as well. Here's just a quick study that we did internally to test all our flow cells. What we did was we soiled them with polyclonal antibody and let it sit for 24 hours. Then we flushed each one with 0.5 molar hydroxide for 30 minutes, and then took a swab at each of the locations pictured below. In every case, the result of the TOC testing was below 500 parts per billion, so we felt very comfortable that this flow cell design is cleanable. Now, you would have to do your own cleaning process and your own cleaning validation, but you can feel pretty confident that your standard cleaning processes should be adequate for these flow cells. And finally, we're committed to your success. The team at Repligen is here to help you. Our goal is to make sure that you have everything you need for this equipment to be successful. We can provide validation, service, and support. So in addition to IQ/OQs that come with the system, we can also help you design your methods, help you validate your analytical methods, help you with documentation and SOPs and data analysis. We have an entire applications team that's devoted to having you be successful, and our support team as well. So please reach out to us and use us as a resource. So in summary, the goal here is to provide actionable data for quicker process understanding, quicker design of experiments, reducing process development time, The system is very operator independent, so it allows for simple scalability, process transfer, easy from development to production, and then easy use in GMP because we have all the certs available and the Part 11 compliance software for you. So with that, we'll go back to Wes for some questions and have some discussion. And so, yeah, we're open to your questions. Thanks.
Okay, Ramsey, as expected, with such a cool technology and so many different applications, we have a lot of questions. So there's a couple different buckets in here, though. So there's a couple associated with integration, and we have one that got the most upvotes, clearly by a long shot. We have 5 upvotes for can the Flow VPX can it be integrated with the KR2i? So let's start there.
Yeah, I think this one warrants a little bit of discussion because we have a lot of cool things in development currently. So the current state of affairs right now is the Flow VPX and the KR2i can be connected and can communicate to each other based on an analog signal that's sent from the Flow VPX. That's what can happen today. You can call me or my application team and we can come to your site and help you do that. That way you'll be able to get the concentration signal onto your KR2i. You'll be able to stop the process based on concentration, and you'll be able to set alarms based on concentration from the Flow VPX. Right now, we're doing a development project to integrate the KFCOM software with the Viper software. The Viper software is the one for the Flow VPX, as you saw, and KFCOM2 is for the KR2i and the FS15. Those two will communicate to each other via OPC UA, which is a standard communication protocol. We'll have a single integrated software that can run both instruments or both systems. And then you'll be able to, like I showed on a previous slide, control your system based off of concentration. For other external companies that make systems, we can connect via OPC UA, analog or digital. So you can tie into things like Delta V, Siemens SciPat, or Unicorn if you'd like. So there's other ways that you can do it, also out of the box with the VPX.
So Ramsey, you mentioned the FS system. So the KR2i is a very well-known small-scale TFF system, and then we just launched 2 FS, like with Quattroflow pumps. Do you have a preference, or is there any advantage or disadvantage between integrating with the KR2i platform versus the FS platform?
I think regarding how the software connects, initially we'll be working on the FS15. That one is really geared towards mAbs, and connects nicely to the VPX. But the KR2i will be fine as well. So I think currently my preference is on the FS side, but I think in the future we'll have KFCOM and Viper talking to each other really, really seamlessly, and should work on both systems.
Okay. All right. So let's skip down to, we have another question on integration, and then we'll go back up a few questions around applications. Can it be integrated with other process measurements, same as temperature, pressure, and things like that? Can they all be balled into one place?
Yeah, they can. I think typically the way customers will do it is they'll use other external software like Delta V that can pull various process sensor data into a single software, and then our system can communicate via OPC UA to Delta V, and then you can get those different sensor data all in one location at one time and pull it into your automation. So that's right now the preferred way to do it.
Okay. And then, you walked through a couple applications, right? Different applications, but the top voted question, it sounds, from my side, it sounds like it's kind of a general question. It's curious about like what kind of samples it can handle in terms of the workflow from like crude all the way out to pure. So the question is, from which stage of the purification of the protein Can the device accurately measure the protein concentration without interference from other contamination?
Yes. So I
Thoughts on
think
that
that's
one?
a really good, yeah, that's a really good question. The reality is we're giving you the absorbance per millimeter of your sample, regardless of what's in the sample. So you can use it essentially anywhere in the process, even in the upstream. Now, you're not going to be able to get actual protein concentration without some complicated calculations in the upstream, but you can get um, the change in concentration over time. And we're working on an application right now for OD600 on a fermentation process, so we can monitor that fermentation process and see when the final the endpoint is for that process. Now, if you want to get protein concentration, really it kind of starts at the capture step. So once you've done your protein A capture, the protein's purified well enough that you can actually measure an accurate protein concentration. So anywhere from there downstream, you'll be able to directly measure the protein concentration. As I mentioned in my slides, before the column, you can take a reading, and after the column, you can take the reading. And from that, you can actually infer the titer, which has a lot of interference, right? But we've backgrounded out the interference essentially. So I think if you're wanting to get direct concentration without any calculation, it's after protein A. If you want to get an idea of what your process looks like in time, you can do it really anywhere from upstream all the way down.
Okay. All right. Sounds good. Yeah, it's interesting. I think you guys have gotten really creative with how to use it in terms of delta or delta absorbance or absolute absorbance. It's I guess you're doing a lot more with it than I think first meets the eye.
Yeah, it's
All right. So
possible because of the slope measurement. The slope measurement gives you a little more flexibility, and it averages out some of those interference that you get from weird samples.
yeah, yeah. Back to integration. Kevin asks, how does Viper handshake with KFCom? Maybe go back to that one again, because I do think in modern technology, the software plays a huge role in its application.
Yeah, like I mentioned, both softwares have OPC capability on the back end. So we directly communicate with KFCom via OPC. So in the future, probably around Q4 this year, we'll have an integrated version of the software that you can buy and get on your system to control both units, both systems at the same time, and actually have that concentration control TFF process.
Okay. Then we have a couple questions around capabilities, right? Luke Walker asks, is there a flow rate limit that the instrument can pick up and still operate?
Yeah, so there was a slide that I showed that had some flow rate limits. It depends on the flow cell that you're operating. Also, pressure is really the key factor there. So we want to keep the pressure under 80 psi. If you're doing that, the flow rate limits are 1.5 liters per minute for the 3 millimeter flow cell, 20 liters per minute for the 10 millimeter flow cell, and 160 liters per minute for this 22 millimeter flow cell. And then we do have capabilities to make larger sizes. So don't hesitate to contact us if you're looking at a larger size, because we can make custom flow cells up to about 2-inch pipe size.
All right. Those numbers sound really well within like standard operating conditions. And then I'm not gonna try and pronounce the name, so I'm just gonna read the question. With the Flow VPX, can we screen different wavelengths automatically? Do you have multi-wavelength capability on the Flow VPX?
Yes, we do. Yeah, absolutely. So we can go from 190 to 1100 nanometers. I didn't present on this particular application, but there is an application where you can use multi-wavelength read during a polishing step and look at the spectral differences during polishing to get an idea of the impurity content of your process pool. This is something that was done at Karlsruhe Institute of Technology a few years ago, and a professor named Jürgen Hubbeck, who published on this a couple times. Those papers are available if you look them up. But But yes, you can definitely do multi-wavelength full spectrum scans in real time. Now, the trade-off is time. It takes a little bit of extra time to do that kind of measurement, but it absolutely is possible and gives you a lot of information about your process.
Okay. All right. And then we do have several questions in the background, but we've got one more that's been upvoted. So we're going to take the last one and then we'll wrap up. Again, if we didn't get to your question here in this session, we'll respond to you by email. So Ramsey, last question, back to software, but not technical, more on, I think, just the implementation side. Is there additional and or recurring licensing costs for the software?
No, there's no additional licensing cost for the software, though. It's often that our customers will get a PM service with our instrument, and in that PM service includes software upgrades. So I would highly recommend to get PM, annual PM with the system, because then we can bring everything up to date. We can also check your system, make sure it's working properly, and then make the correct modifications to ensure that the thing will run forever accurately.
All right. So Ramsey, I think with that, I'm going to thank you for the excellent presentation, tech chat, answering the questions, all of that. Thanks for taking the time joining us today. Thank you everyone for joining. Again, see the questions. If we didn't get to your question today, we'll wrap it up with an email response to you, and we'll look forward to seeing you on the next webinar next month. Take care.